Thank you for that kind introduction, Luis. I really love giving this sort of talk because I'm going to make my best effort to avoid getting into the nitty-gritty details and showing you lots and lots of data, which means you sort of have to take my word for things and believe what I have to say. Secondly, Luis, I will be asking you for a favor someday. Who knows when it will be, but hopefully you can return the favor for me. It really is a pleasure to be here, and I also, looking at the audience, have noticed how well Texas scientists really clean up. This is quite an impressive group sitting out there. So as Luis described to you, my laboratory is interested in the neural circuitry that controls movement. And one of the key areas of the nervous system that contains the important neurons for controlling movement, not the only place, but one of the key areas of the nervous system is, in fact, the spinal cord itself. And the spinal cord contains many of the neurons that control the very kind of subconscious sorts of movements that we make, especially, as an example, the kind of movement that you might make if you were to touch a very sharp object or a very hot object and to withdraw your hand. That requires using lots of different muscles and is a very sophisticated kind of response, yet is something that happens without very much conscious input from your brain. A few milliseconds later, you might realize, oh, I just touched something sharp or touched something hot, but it goes to the point that the control of movement happens at many different layers or levels within the nervous system, and the spinal cord is one of those important areas where it is controlled. We've been interested in the circuitry that controls movement from the standpoint of trying to understand how it is assembled during development. And we regard development as the phase prior to birth, the embryogenesis of the nervous system, as well as some of the early postnatal stages when there's still some refinement and improvement that is being made to the nervous system to help control things like movement, as well as many of the other things that the brain performs. How is this relevant for human health? Well, some of these things are probably quite obvious to you, but there are a variety of things that have a direct impact on the performance of movement. Those include things like spinal cord injury, but also a whole array of different diseases, which I've listed some of them on these slides. So we're studying the early assembly of the nervous system. How does this relate to some of the diseases? Well, perhaps somewhat indirectly. By the end of this talk, hopefully you'll see that there might be more direct connections than we had previously understood. But part of the logic for our work on embryonic formation of the neural circuitry is that if we can understand some of the principles that nature is using to actually put the nervous system together, we might be able to use those principles again to try and restore function or regenerate parts of the nervous system. So we're really trying to get at some of these deep secrets that nature has with regard to building a nervous system so that we might be able to use them at later stages to treat some of these diseases and injuries. So Dr. Huber already has alluded to some of the information on this slide. I would like to point out that one of the main questions that we often deal with in my laboratory is what I've listed here as the complexity paradox. And what I really mean by that is that the number of cells in the brain and the spinal cord that are neurons that are absolutely critical to the function of circuitries that underlie various behaviors, such as movement, are immense in number, 100 billion cells. These cells are highly interconnected with one another. This is how they relay information to one another. It's these interconnections that really create a nervous system into a highly functioning system that allows us to remember things, to respond to our environments, to link a beautiful sight or beautiful odor with particular emotions. So these connections are really at the fundamental function of the brain. These are big numbers, 100 billion cells, 100 trillion connections. These are only the kinds of numbers that somebody who works in the oil industry might be able to understand in the context of profits. For us, we deal with smaller numbers in my lab, and the smaller numbers represent the number of genes in our genome. And so over the last decade or so, tremendous progress has been made in terms of genome sequencing. And what we now believe is that there are on the order, this is a rough estimation, that there are something like 25,000 genes. So this was a great day when we came to the conclusion, 25,000 genes. It was also, it raised this paradox that I'd like to point out, which is how, with a relatively limited number of genes, can you build up something as sophisticated as a brain with all of these cells and all of these connections? And so it has been this particular paradox that has been driving our research to try and understand gene function in much more detail, to understand how you might be able to create this complexity with what we regard as a relatively small number of genetic components. So first I would like to take a moment here to point out some of the neuroanatomy that is going to be relevant to some of the genetic pathways that I'm going to talk about in a moment. So the neuroanatomy I want to point out to you is something that has been, I would say, moderately well understood for over a century now. And so on the left here, this pointer is a little bit hard to see here, but on the left is a drawing that is over a century old, done by Ramon Cajal for the neuroscientists in the audience, in which he has traced some of the key neural pathways that are associated with the control of movement. So some of our volitional movements are controlled by neurons that are located in the cortex, specifically the motor cortex. These neurons send long processes down through the brain stem, and into the spinal cord, where they then terminate on neurons that are located in the spinal cord and control the function of those neurons in the spinal cord. There are also inputs coming from this area of the brain, the cerebellum, to help control movements as well. A lot of these connections from higher order areas of the nervous system, brain and cerebellum, are really important for volitional control of our movements, and also for some of the things that are uniquely special to humans, namely our ability to use our fingers in very precise ways. So most other species do not have the ability to this very fine, exquisite motor control in their hands. And the neural circuitry in other organisms, while highly related, differs a little bit in the context of what's going on in the motor cortex and the connectivity within the spinal cord. So once inputs are received into the spinal cord, what is the next step in the circuitry? Well, there are cells that are located within the spinal cord. It may be a little bit hard to appreciate this, but this is supposed to be a cross-section of the spinal cord down here. And these neurons within the spinal cord, some of them are making local networks of circuitries within the spinal cord, but then there's an output cell that has to relay all of this information out to muscles. And that output cell is called a motor neuron. So this is a relatively easy name to keep track of here. These are the cells that die in neurodegenerative diseases like Lou Gehrig's disease, motor neurons. Now, motor neurons are somewhat special in the nervous system for a couple of reasons. One, they're immensely big cells. Their cell body resides within the spinal cord, but they have one long process that goes all the way out of the spinal cord and contacts muscles. So if you're Shaquille O'Neal, you have motor neurons that extend from your spinal cord to the tip of your foot that are 3 feet long or longer. And this is the world record holder for cell length. Big cells, and these cells make direct connections onto the muscle fibers to control movement. So that's what's meant to be shown in this schematic. Remember now, the cell body for a motor neuron resides within the spinal cord. It then sends a process we call an axon out of the spinal cord. This occurs during embryonic development. The cells are initially created in the spinal cord, and then they grow these very long processes during embryonic development out into the periphery where they form synaptic connections with muscles. So how do we study the development of these neural circuitries? Well, I maybe should make one additional point here. So I've just told you that motor neurons reside within the spinal cord. I should also add a couple of other pieces of information at this point. Firstly, there are approximately 650 different muscles in our bodies, and the connectivity between motor neurons and the muscles is done in an extremely precise way. There is no miswiring that occurs during embryonic development. The learning process that is occurring in children after they're born to learn how to walk is not a reorganization of these connections between motor neurons and muscles. It is a different type of learning that is taking place right from the outset of embryonic development. There is a hardwired genetic process that is taking place to ensure that the right motor neurons have connected to the right muscles so that when instructions or signals are sent to motor neurons, the right muscles are contracted. Now, this is a slightly more complicated problem than I'm even trying to describe to you, because not only does the motor neuron have to fire an action potential to tell the muscle to contract, but the relative firing patterns of motor neurons going to different muscles need to be precisely coordinated with one another so that muscle contractions occur without another muscle on the other side of the joint opposing the force, and so that the relative timing of different muscle contractions is precisely controlled. So there's a hardwired genetic process, and that genetic process is really very carefully specifying during embryonic development the growth of motor axons out of the spinal cord and toward the appropriate muscles, whether it be muscles in the arms, the hands, the legs, the feet. I think you get the point. So the system that we have used to try and understand this wiring problem, now focusing on the wiring problem that motor neurons have in trying to find their appropriate targets, has been the mouse. So there are several reasons why we use the mouse as our model system. One is that there is an immense homology between the genomes of humans and mice. This might be a bit disappointing to you, but we don't have many more genes than a mouse has. We obviously use our genes in a slightly more sophisticated way because we can do few things that mice can't do. But we're maybe not all that special compared to mice, because many of the same kinds of locomotor things that a mouse performs, humans also perform. And this is one of the assumptions that we make, that studying mice and the neural circuitry that controls movement will inform us about what is going on in the human nervous system. So in this particular movie, I want to show you a genetic example of a defect that alters the way that a mouse moves. So here's a mutant mouse, and hopefully you can see that the way he moves is different than the first example of the mouse, the first mouse example. Maybe I can play that one more time if I do this. So this is a normal mouse. You take a look at his back legs as he runs across the pole, and here's a mutant mouse. And watch how he hops along the pole like a kangaroo. Now, we didn't spend a lot of time training the mice to do this. In fact, this recording was done here in Texas with one of Luis's colleagues, by one of Luis's colleagues. And this is a genetic mutation that predisposes these animals to use hopping as a way to move rather than to use alternating limb movements. And it was these kinds of observations that suggested to us that we might be able to use mouse genetics as a way to begin to understand some of the genes and the genetic processes that are involved in building up the neural circuitry that controls movement. And so now I will show you just a little bit of data. So this is an example of a transgenic mouse that we made. This is a cross-section of a mouse embryo shown here on the left. And what we have done is to make a mouse in which we have expressed a green fluorescent protein selectively in the embryonic motor neurons. And so the green labeling that you see represents the cell bodies of the motor neurons that reside within the spinal cord. You see them on both sides of the spinal cord because this is a bilaterally symmetrical organ, like many of our organs, not all of our organs, but many. And these cells shortly after they've been created in the spinal cord, have now begun to grow very thin axonal processes out into the periphery. Unfortunately, we don't have the level of resolution with this particular image to see the individual axonal processes, but there are enough of them coming out of the spinal cord, and they're close enough together that you can see this long line of green labeling here. What different motor neuron types are doing are making very selective decisions about where they're going to grow. So they first make a decision to grow out of the spinal cord. This is different than most neurons within the spinal cord that do not exit the spinal cord. They then reach a point right outside of the spinal cord where they make a decision about whether they're going to turn to innervate a group of muscles that will form along this portion of the embryo, which will eventually become some of the so-called axial muscles, which will support the posture of the animal. And so this is a subgroup of cells that are doing that, whereas others will continue to grow, and this represents the embryonic limb bud for the back legs, and they will continue to grow toward the limb, and there they'll make a decision about whether to go one direction into the limb or the other direction into the limb. So each of these branches is comprised of processes from different motor neurons, and they are making, as I said, very, very selective guidance decisions about how they're going to navigate. Now, this isn't an actual blow up of the tip here, but is an illustration. If we could really blow this up adequately, what you would find on the tips of these neurons is a navigational device that we in neuroscience call the growth cone. The growth cone is a highly dynamic structure. It has a very dynamic cytoskeleton within it. There's filamentous actin, which has been labeled in red here, which can be readily reorganized on the tips of these neurons, and then microtubules behind that, supporting and stabilizing the axon shaft. And so what is going on during embryonic development is that there are receptors that are embedded within the membrane of the growth cone. Those receptors are detecting signals in the periphery that help to dictate whether the growth cone is attracted or repelled from particular signals. And in this illustration, what I've tried to indicate is that there's a positive signal whose concentration is greatest up in the upper corner here, and that the way that the growth cone will respond is to build up its cytoskeleton in a way that promotes growth toward that signal rather than in a different direction. So we've been very interested in what are the signaling pathways that help to control this very selective growth of axons that underlie the very precise wiring within the nervous system. In part to learn exactly how motor neurons do it, but also in part to learn how, more generally, the entire nervous system is wired up, because the principles that are used by motor neurons are likely to be very similar to the principles used by other parts of the brain. So here's where I want to summarize many years of very hard work by very talented people in my lab and tell you some of the rules that we have discovered, we think relatively general rules, that help to explain how relatively small numbers of genes might be able to contribute to the wiring of a very, very sophisticated structure. And there are a couple of principles that I'm trying to outline here. So one of the things that we have discovered recently is that there isn't a one-to-one relationship in terms of neuron growing to target and the target producing something that is attractive for the neuron, but rather it's using combinations of signals, combinatorial codes, much like we would use an alphabet to form many different types of words. The other thing that we have discovered is that these signals, these combinations of signals, have to be tightly aligned in terms of their position within the embryo and the timing with which the cell receives them. And so we've discovered that if you find two signals that can promote axon growth, but those two signals are separated from one another in time, that you can get small responses by these signals, these secreted factors, we call them ligands, that bind to receptors embedded within the membrane of the growth cone of motor neurons, will produce small responses that might lead to small amounts of attraction. But that the way that the system has built itself up to have even greater fidelity is to require that the two signals be very close to one another in space and close to one another in time, so that the receptors actually perform physical interactions with one another that help to amplify the signaling and lead to a much better signal output, which helps to resolve some of the signal-to-noise issues. And it's these kind of combinatorial principles and combining or coordinating the signaling in what we've called a coincidence detector that helps to enhance the fidelity of axonal growth. So the strategy that we have taken to try and uncover new genetic players in the system is a genetic strategy in mice. And this has been greatly facilitated by high-throughput sequencing technology. So some of this information will now be perhaps most appropriate for some of the real experts in the audience. I'll try not to belabor the points here. What we have done is to create a mouse line in which we took a regulatory element out of a gene that is selectively active in motor neurons. We linked it up to the green fluorescent protein, and we revved the system up so that this green fluorescent protein is produced at very high levels in motor neurons, so that we could dissect out embryos from pregnant mice and look with a very low-power microscope at the way the axons were growing into the embryo. And so this may not mean a lot to you, but this is a very, very stereotypical pattern that forms with very, very great precision. And this is sort of a reference point for how the motor system is wiring itself up during embryonic development. So then we used a mutagen called ENU to create mutations in the genome of these animals, and then did crosses to try and find mutations that would alter this particular pattern of axonal growth. And the way the numbers work here is that in our particular genetic screens, we screen through about 250,000 mutations in the mouse genome to find 6 different genes that are important for axonal growth. Because of the interest in deep sea technology in this room, we named our mutants after explorers who often got lost on their way to their ultimate destination. So our mutants were called things like Magellan and Columbus and De Leon and so on. These represent mutations in the genome that alter the way that motor axons are growing toward their muscles. And this may be subtle for some of you, but these represent the ventral roots. So the neural tube or spinal cord, one half of it is shown here, and the axons are coming out of the ventral roots on their way to targets. And what happens in the Columbus mutant is that kind of organization completely breaks down, and so now what you begin to have is something like a hairy mess rather than a highly organized segmental pattern of axons. So using high-throughput sequencing technology, we mapped and cloned the mutation responsible for this particular mutation, and this particular mouse line we called Columbus, but it's a mutation in a gene that is called presenilin, or abbreviated PS1. For some of you in the audience, you may already know this gene. It is a so-called protease. It also goes by a more technical name, gamma secretase. What is a protease? It is equivalent to a molecular scissors that cuts up other proteins. And what is special about presenilin? Well, a variety of things, but one of them is that this is a protease that resides within the membrane itself, where it has very close interactions with some of the proteins that control axon navigation. And the mutation that we isolated was one that destroys a key part of the protein, preventing it from being expressed so that the catalytic activity has been lost from the protein. So if you look in the midline of the neural tube, one of the things that we found is that motor neurons, rather than growing axons out the ventral roots, out toward muscles, were now somehow attracted to the middle of the spinal cord. This structure is called the floor plate, and so you can begin to see green labeling in the middle where you normally do not have labeling. We then did a search to try and find why are these neurons now inappropriately attracted to the middle of the spinal cord rather than wanting to leave the spinal cord. And we discovered that there is a rather well-known axon guidance molecule called Metron, which under normal conditions does not attract motor neurons to grow toward it, but in our mutation, it now does attract motor neurons to grow toward it. And so this is a ligand whose receptor is already known. It's a ligand, as I said, Metron, and its receptor is known as deleted in colorectal cancer, or abbreviated as DCC. And in geneticist terms, what we believe is going on is that presenilin, this protease that we found to be mutated in the Columbus line, is inhibiting this signaling interaction from promoting the attraction of motor neurons toward an inappropriate source. So it's trying to block an inappropriate response. Now, why, you might ask, would nature devise such a system? If it doesn't want to grow to the midline, why does nature put a receptor in motor neurons that could even lead to such a defect? And nature doesn't always pick the most economical solution to its problems. So I'm seeing that my time is up, and so I'll try and wrap this up here in a timely fashion. So one of the things that we believe is going on is that, in a way, as a mechanism to help diversify the function of different genes, is that the timing with which the genes are active is tightly regulated. And in this particular example, so I was mentioning that you might say, why would a motor neuron, or ask, why would a motor neuron even express a receptor for something that might lead to its growth to the wrong place? And one of the things that we have noticed is that this ligand Metron is also produced by cells that form the axial musculature and parts of the limb. And so we think that part of the logic behind this is that the presenilin gene is helping to ensure that motor neurons will be able to respond to this signal, but only after the motor axons have gotten far enough out into the periphery so that this place where Metron is present, which is involved in wiring up other neuronal classes, can be ignored. So we think it's part of a kind of a binary switch. Give me enough time to get away from something that I don't want to be attracted to, and once I get out into the periphery, then I want to become highly attracted to that peripheral signal. I want to conclude by actually talking to you briefly about what we think or what we know are some of the functions of presenilin. And so when we discovered this gene, it was actually quite a big surprise to us, because this was already an extremely famous gene, because it has been associated with familial forms of Alzheimer's disease, a neurodegenerative disease that strikes people who are older, not something that had been associated with embryonic development. And one of the things that mutations in presenilin has been known to cause are inappropriate protein plaques that form within the brain of Alzheimer's patients and lead to neurodegeneration. And so mutations in presenilin in humans have been known to lead to this misfolding of proteins. And so I want to just conclude by saying that what we have discovered is a somewhat surprising link between neurodegeneration and presenilin, and also the uses of presenilin, if my pointer will work here, nevertheless, some of the functions of presenilin in other contexts, such as neural wiring, and it even leads to implications about how we might be able to control presenilin activity to help regenerate parts of the nervous system. And so with that, I just want to conclude by pointing to the 3 postdocs who've done the work I talked about, our collaborators, and our funding. Thank you very much for your attention.